Right, we're still processing on preparing our samples for PCR. The next, having extracted our DNA, we've got that ready, haven't we, in the correct tubes, in the PCR tubes. We now need to make sure that we're going to add either a master mix formulation or a PCR bead method. Now Mary can explain to you the difference between those. Am I right in thinking that we're going to do the first one, the master mix?
We're going to use master mix.
Okay.
And there's a reason for that.
Okay, right.
Okay, shall I explain? Right. Both master mix and PCR beads contain the enzyme that does the copying of the DNA.
Okay, right.
It's called Taq polymerase.
Taq polymerase, yes, okay.
And Taq polymerase can work at very, very high temperatures.
Sure. I mean, there's a story behind the discovery of that, which we won't deal with at this point.
Yeah.
That could be a supplementary issue, but Taq polymerase was the key. The key, wasn't it, for this whole process? Yes.
Yeah. So Taq polymerase can work at temperatures 95, 96, 99, doesn't matter.
Yeah.
It's quite happy at those
sort of blows some of the enzyme theory out of the water a little bit, doesn't it?
Yeah. Everything's got edges, now they both contain that enzyme, and both of these also contain the buffers, which give exactly the right pH for the enzyme to work, and also enzyme activators and things like that.
Right.
So that they both contain that, and what they also both contain is they contain the building blocks for DNA, the nucleotides.
Oh right, okay, so that's all, hence the phrase master mix, I suppose, then, because everything is inside that mix and we just need our DNA sample to be added to that, then to be able to amplify.
That's right.
Okay.
Now the difference between them is basically that PCR beads are dehydrated. So in one PCR bead you have all of the ingredients that you need, apart from the water, that will actually do the copying of 120 microliter sample of DNA.
Am I right in thinking that we have a dehydrated version of master mix? Is that what
That's
this is?
Right, yeah.
Okay.
Now, if you buy in PCR beads, they're very easy to manage with your class. But once you've opened the pack of PCR beads, you must use them with that class. They won't keep.
Right. Okay. And is it one bead per sample?
One bead per sample.
Okay,
yeah.
Right.
For master mix, you have a tube this size, and it contains enough master mix for 32 samples.
Right.
So what you do when you get it in is you aliquot it out into separate containers.
Yeah, because the last thing we'd want is our student contaminating the whole perishing lot. We'd want to have those student-specific little containers and some technical work in the background to do that.
So you aliquot enough for a class to use, and you may not need to use all of this.
Right.
What you don't need to use, you put it in your freezer, and it'll keep until you just need to defrost it.
Right. So I think that's just down to personal circumstances, isn't it? Whether the beads or the master mix is an option for your group.
Yeah, it's basically you make your choice.
Okay. How about the water? Not tap water, obviously.
What you will do is we'll talk shortly about primers.
Yep.
And primers already come in a liquid, and you basically add the primers, and you may need to add a very small amount of sterile water to your beads.
But we'll come to that a little bit again.
We'll come to that later on. If you need to do that, we're going to be using master mix in our process here today.
Okay. Right, thank you. We're moving on in a moment.